technique is most promising technique in the near future because this is the very this is the only technique you can modulate the different stimulation with different region without any surgery. So this is very promising. And personally, I'm pretty sure this is very useful technique in the near future. So and then I believe that everyone in this place will contribute a lot to save the world. So this is very really exciting. So to make that happen, many people try to stimulate the brain with ultrasound in many animal models, for example, mice. This is common animal models. But this brain is very closed system, so it's hard to see the neural activity directly because of this is closed system. So instead, people try to use the indirect method to measure the to see the neuromotion effect. For example, people try to see tail movement or EMG or head movement while ultrasound stimulation. So this is very useful. And some people try to use the metal electrode, and then they implant this electrode into the different region, and then they overlap it with the beam path. So they try to use many people try to use that approach to test the ultrasonic neuromodulation effect. This is very useful, but the problem is that because of the acoustic mismatch at the solid substrate, the ultrasound can hit the solid substrate, and then it can make some movement with a vibration. And this vibration makes some the kind of the motion artifact, at the electrical artifact, and also it can push the cell directly. This vibration can push the tissue directly, and they cause some another artifact in behavior in animal behavior. And also, some people try to stimulate the brain, small animal. So people try to stimulate here, but people didn't see any local target activation. But instead, people saw some open target activation effect here, especially we saw some very strong activation at the auditory cortex. And then auditory cortex activation of this region also elicits some motor responses. So there are some, this is the undesirable results with ultrasound, but this is very important finding for the better fun. So this is very important. But after report this story, many people come to me and then talk to me and then ask me, hey, I think this ultrasound is not real. This is real, Sangjin? But I think this is real because we saw some very specific results. For example, people saw some target responses using the deafening mouse, and then people saw some tactile sensation, and then some change after finish communication. So this result is not able to be explained by auditory artifact. So this is the real effect, I think. But as you know, there are some, if you apply the ultrasound, sometimes the brain modulation effect is very weak or minor. So we address this limitation, and then with that, we need some more fundamental study to make the better fun technique. So we try to do some fundamental study using some other approaches. So for the fundamental study at the molecular and cellular level, so we try to use. I didn't use the in vivo approach because of the auditory artifact, and then it's hard to see single cell level effect using this in vivo setup. Instead, we developed some 2D platform where especially we touched the neuron onto the minor film, which is the acoustically transparent substrate. And then we use the low frequency ultrasound because especially 300 kHz, this is widely used in the human applications. So we use this frequency, and then we express some GCAP sensor into the neuron to see calcium response while ultrasound stimulation. And also we try to develop some 3D platform as well instead of this slice, because this plug-in 3D platform is much stable, and then it's much better to optically access to the cell responses in more better than slice. So we try to use this 2D and 3D culture system for the fundamental study at the molecular and cellular levels. So we try to simulate a cell with many kinds of parameter, and then we finally see yeah, it happens always. Yeah, there's some calcium activation while after ultrasound stimulation, and then this you can see this is calcium response after ultrasound stimulation, and then this calcium response is very clear if you use the higher or longer sonication parameter. So this is very clear neural excitation effect. And then this is the this stimulation we use for that. We try to use those continuous wave for the neural stimulation, but we also try to use the pulsed wavelengths to stimulate the brain, so it was cells. And then we saw very clear responses, but there's no big difference between the calcium response. So you can see here, there's calcium responses, but there's no big difference between the continuous wave and the pulsed wave. This is only 2 parameters, but there's no big differences. Instead, we saw some very interesting differences. For example, if you use higher frequency, for example, 670, we saw some big, little bit bigger responses. And then in case of 900, almost 1 megahertz. In this case, it's hard to see very clear neural responses. So maybe I think there's some clinical point between the 670 and the 900. So for the neural excitation, we found that the ultrasound intensity and duration and then frequency is very important. And then if you look at calcium response more detailedly, so you can see here there are some time delay, response delay. There's some delay before the calcium elevation here, and this is the almost around 100 or 200 millisecond delay. And then this delay is already happened, but you can see this delay is decreased if you use a higher intensity of ultrasound here. So we think something happened inside of cell within this time delay window. And this calcium response to the ultrasound stimulation, you can see clear response every time. And then we we added some TTX, sodium channel blocker, and then we saw a big reduction in calcium responses. But we still see, still see the calcium responses, which means there's 2 steps. First one is calcium enter into the cell directly after ultrasound stimulation, and then calcium enter again after depolarization. There's a 2 step. And also we try to block every synaptic connection using some chemical synaptic blockers, and we still see very clear responses even after synaptic blocking. So which meaning each neuron responds to the ultrasound on its own. So there's no any synaptic effect. And also we try to split the cell using 3D platform. So we try to use a 3D neural network inside the collagen. So we mimic the brain stiffness using this collagen, and then we stimulate the cell, and we still saw very clear neural responses. And then we look at the time delay before calcium responses, but there's no any big difference between the 2D and 3D culture, which meaning the previous calcium response at the 2D, from the 2D is not some artifact because of 2D. So so calcium response is actually rare. And then we also found that if you apply ultrasound, we saw some calcium release from the ER too. Here, this is ER. So we saw calcium release from the ER, but this is very, very small. So, and then we found that this calcium release from the ER is not the major mechanism of the ultrasound inflammation. Well, this is a new finding. So, but what is the mechanism? So we saw some neural activation effect using ultrasound. It's very exciting, but we don't know the mechanism. So we try to find the mechanism using our platform. So as you know, there's two categories, the biophysical interaction of ultrasound. So first one is the thermal, the other one is non-thermal. So we try to test all of them using some optical or chemical or genetical method. Oh, this is different. I see. So we try to test temperature effect. So for that, we try to express some temperature sensitive protein, for example, M-cherry, and then we stimulate cell using ultrasound. But we don't see any signal perturbation, which meaning there's no any significant temperature increase. And also we try to use some hydrophone, so we try to measure the temperature increase near the cell. And then we saw some calcium temperature increase, but this is very, very small. So and then there are a lot of computational model study, and then they also conclude that temperature effect is very, very minor in this parameter. So we rule out the temperature effects for the mechanism studies. And next, we try to study the bubble effect, for example, cavitation. So as you know, ultrasound can make some bubble. And then this bubble can push the cell membrane, and sometimes you can make some pore, and then you can make some neutralization effect. So we test this hypothesis using by simply using the degas media. So we degas the media, and then we stimulate the cell, and we still saw very big response, very clear, which meaning this bubble cavitation effect is not unlikely. And also, if you stimulate the neuron, you can see the calcium responses here, and then they go to baseline after a few seconds later, and then this always happen here, which meaning there's no any ionic unbalance because due to some membrane population. So this is the so we rule out the microbubble effect as well. And then also we try to test the intramembrane cavitation theory. So in computational model, people predict that maybe ultrasound can make some microbubble inside of the membrane, and it causes some activation effect. So we wanted to test this hypothesis too. So for that, actually because we use the degassing media to eliminate any bubble formation. So I think this is the unlikely, but we try to want to make sure. So we try to we wanted to see some bubble formation near the cell or intramembrane to using any very high speed imaging technique. So, but this is cell body, but we're not able to see any membrane, any bubble formation and then membrane deformation using this technique. And also, we try to use the voltage sensor. This is the voltage sensor. We express this sensor to see the kind of deep hyperpolarization. So computational model predict that if we apply the ultrasound, this intracellular cavitation cause the kind of deep hyperpolarization before excitation. So we want to see that kind of deep hyper using this voltage sensor. So after we stress this voltage sensor, we apply the ultrasound. Then we see a very clear dual excitation effect, but we don't have to see any deep hyper effect during the response delay. So we rule out this intramembrane cavitation too. But instead we found very interesting. For example, this is the spontaneous activity. So this is the spontaneous activity of the voltage imaging, and this is the neuron actually can make the kind of activation even if there's no any calcium extracellular spaces. So even if there's no calcium inside in the extracellular space, they can make some activation. They can make some action potential. But in this condition, we apply the ultrasound, but we're not able to see any neural excitation effect in this condition, which meaning calcium influx is very important, and that this calcium influx is first event after ultrasound stimulation. So this is a very big finding. So calcium is very important. So okay, calcium influx through the swim channel is very first initial event. So maybe we think without maybe mechanical force of the ultrasound stimulation, maybe radiation force may stretch the membrane, and then it can make maybe this stretching effect may activate some ion channels, and then we saw some. That's why we saw calcium influx. But as I told you, we're not able to see any that kind of membrane stretching effect using ultra high speed imaging. So instead, we try to target this cytoskeleton, because if there is some mechanical deformation at the membrane, there will be a mechanical deformation at the site. There will be deformation at the cytoskeleton as well, because they connect to each other. So we try to target this cytoskeleton to study that kind of hypothesis. So for that, we try to destroy the actin structure using some specific chemicals, and then as you can see here, and then we apply ultrasound, and then we saw a big decrease in the calcium responses. So which meaning there is mechanical transduction through the actin. So this is the big finding. And also after actin deformation, we found that there are many mechanosensitive channel opening. So this is confirmed by gadolinium. This is global inhibitor of the many mechanosensitive channel. So you can see a calcium decrease after GD. So which meaning, so again, ultrasound stimulation may stretch the membrane. It causes some deformation of membrane and cytoskeleton, and then it causes some activation of mechanosensitive channels. So this is very exciting. So we know, so we found the mechanosensitive channel is very important after some deformation. And then we want to know which ion channel is actually activated by ultrasound. So there are a lot of mechanosensitive channels we can test. So first, I try to use some chemical inhibitor. If there is potent and very selective inhibitor, if there is, I use that chemical blocker. But if there's no any very specific potent chemical inhibitor, then I try to use the CRISPR technique to target specific ion channel. And then we found that a couple of mechanical sensitive channel, for example, TLPP1, 2, PIZ1, TLPC1 is activated by ultrasound. And also some of calcium activated amplifier channel, for example, TLPM4 and the T-type calcium channel is activated by after this channel activation. And then to put a double compound, so we trying to overexpress a couple of ion channel, for example, TLPC1, TLPC2, and then TLPM4. So we overexpress this channel and then stimulate the cell, and then we saw very big higher response at the using the low intensity ultrasound, which meaning our screening is correct. So this is the whole picture of the biomolecular pathway. So if you apply ultrasound, there is membrane stretching, or there is some membrane deformation, and then there is some actin deformation, and then they cause some calcium influx through the voltage mechanosensitive channels. And then after tens of millisecond delay, we need some tens of millisecond delay for the calcium accumulation. And then if calcium accumulation is enough, then this calcium activates the calcium sensitive amplifier channel, for example, these channels. And then finally they activate the voltage calcium and sodium channel to make the bursting activation effect. So this is the molecular pathway. We found that. This is very exciting, but this is not the end. There is a lot of open questions. For example, I said maybe there is membrane deformation, and then there is actin deformation too. This actin deformation is clear, and then there is we screen which channel is active. We found some mechanism sensitive channel, and they all are membrane stretching sensitive. So I think there is membrane stretching effect after ultrasound stimulation, but using our data, for example, the high fast and the high resolution and the high fast imaging technique, we not able to see any kind of membrane stretching effect. And then I don't think the ultrasound can stretch the membrane at the single cell level, because the wavelength ultrasound is much bigger than cell size. So I think this is not the single cell level, single cell membrane stretching effect. Instead, I think maybe ultrasound push the local network, and this, because of this local network pushing effect, there's some nanoscale membrane stretching effect at every cell, every axons, and then that kind of nanoscale deformation can actually activate that kind of nanoscale deformation may initiate calcium influx for the utilization. This is my hypothesis. But as I said, there's still there are so many many parties still unclear. So for example, we don't know the precise force to activate the cells, and then I think this the membrane stretching effect may be nanoscale event. So we need to use other technique to capture this nanoscale event. And then there's a lot of open question. For example, there's a long-term effect. For example, inhibition effect after ultrasound stimulation, but we don't know why. And then there will be we don't know if there is some cell type specificity because neuron has some different cell shape, and then they are size of cell bodies that they all different. So maybe there will be different mechanical effect according to the cell type, but we don't know yet. So there are a lot of homework yet. So this is very exciting. As a part of biophysical mechanism, we try to target the primary cilia. This is very surprise. So cilia is actually very interesting organelle. This is a multi-sensor, and then every cell, most of every cell express this have a kind of cilia structure. So and then this cilia actually can sense many chemical, and then it can also sense the mechanical deformation and the shear stress, and then also neuron also has some. Neuron also has one cilia at the cell body. So this is very. So we're trying to target this cilia with this hypothesis. For example, if you apply ultrasound, maybe there is some microstreaming effect near the cell, and this microstreaming may cause some deformation of the cilia, or maybe there are some shear stress may stretching the ciliar membrane. And then that mechanical deformation may activate some mechanism channel. This is the channel energy in the cilia, and then that will initiate the calcium signal. So this is the another surprise biophysical hypothesis in our group. So we try to test this cilia using some method. First, we try to express some GFP in live neuron to see cilia structure. So you can see the cilia, say hello with the cilia. And then we try to simulate the cell using ultrasound, and then we still saw very strong exciting effect from both. For example, cilia negative and positive cell, they all activate by ultrasound, and then amplitude of calcium response is almost the same. And then after that, we try to use the long cilia or short cilia. We make the long or short cilia, and then we stimulate the cell, and then we still saw a very similar calcium activation effect. And then we try to see cilia movement while the ultrasound stimulation, and then we don't see any big deformation effect while ultrasound. And then turns out the TRPP2 channel is actually expressed on the cell body, not the cilia. And then every cilia actually connect directly bind to the axon and dendrite to make the snaps. So they are physically bind to the axon. That's why we don't see any kind of deformation even if we use very high pressure of ultrasound. And there's one paper published in very few weeks ago. They also found that there are axon and then neurite axon and cilia is actually bind to each other physically. So our study is actually supported by this study. So with this study we rule out the ciliar effect. So this is very sad, but we can make some another opportunity, for example, sonogenetics. So yeah, I can zoom in. So we try to use the cilia. For example, we can overexpress some specific ion channel onto the cilia to sensitize by microstreaming or mechanical effect by ultrasound. And or we can also bind some we can attach some nanomaterial to make the better physical. We can promote the physical. We can add the nanoparticle to the cilia for the better deformation. So we try to develop some that kind of new sonogenetic approach. For that, we try to use the we make some long and short cilia structure, and also we try to overexpress some specific ion channel to the cilia. So that will be very interesting, brand new sonogenetic brand new sonogenetics project. So you can see very fun sonogenetic study in the future. So in summary, we saw very clear realization effect using ultrasound. So I say this is clear. And then we found we uncovered the biomolecular pathway, and then we ruled out many hypotheses using many approaches, including cilia. But this is great. So this is my end of the presentation. I would like to thank you to Mikhail. He's really great PI ever. And I would like to thank to my friend for the many study, and then thank you to funding source. So this, yeah, thank you very much.
Thank you very much, Dr. Yu, for this nice presentation. We have time for a few questions. Any question? Yes?
Could you please come to the microphone here, then your voice will echo.
I have a question about this model you're using. There are no astrocytes, or there is no microglia involved. Is this neural response in a dish, or is there some microglia, some glia, some dendrocytes involved in the responses?
This is the glia-free platform, so there is no any isocyte and then microglia in this platform, so only neurons.
Any speculation that it might be important?
I think it might be important because the isocyte also has some mechanistic channel, and actually they same with compared to the neuron. So actually we plan to test the isocyte effect. So I think that will be very great project in the future.
Another question? Yes?
So maybe I wasn't mentioning in the beginning, but what the parameters for using study results? And did you vary any specific, parameters?
Yeah. I try to use the mainly I use 300 kHz and 50 watt per square centimeter, and then which is the almost 0.4 megapascal, and then first duration I use up to 500 milliseconds. So this is the best parameter for me. And then we also try to use the different frequency, for example, 670 and 900 something. So we saw some activation effect, but patient response is a little bit different depending on the frequency.
And this is all right?
Yes, yes.
You have any plans on using culture that has like non-functional number of substances or portion methods that act? Because your system is awesome, I love to do condition, but you're looking to solve it non-functional.
There is spontaneous activity, and then I think the spontaneous activity is very important. In my case, if there's no any spontaneous activity, there's two reasons. First, cell will be not healthy, and then even if cell is healthy, if there's no any spontaneous activity in slice or culture, we're not able to see ultrasound responses. So I think spontaneous activity, baseline activity is very important to induce the patient effect.
Well, I have one more question on that. Are there cells that are better, some that are closer to the last and further?
Personally, I tried to test the glass as well. In that case, I saw much bigger responses, and then I saw some selective detachment from the glasses. So I think there are some vibration. This is very clear. Yeah.
What's your thinking of the mechanism of getting a mechanical response that is of frequency and especially the mechanical response at a higher frequency?
Yeah, this is the frequency effect is this is I think if you use very high frequency ultrasound stimulation, the focal length is very small. So I think because of that, the stretching effect area, stretching area is very small too. So because of that, I think the activation effect is very mild. But instead, if you use a little higher frequency, lower frequency, for example, 300 Hz and then 670, the searching area is large enough, and then you can make some there are some clear searching effect. So because of that, I think this is not clear yet, but this is my hypothesis. So I think because of the focal diameter and then searching effect at the net on the network, that maybe cause some different calcium responses. So we need some another computation model to predict some mechanical searching effect.
Okay, thank you very much again. So there was a technical problem with the next presenter who is presenting remotely. So this presentation will be moved to the next session. And now we will receive Ivan Castellanos from the Lab2 laboratory of the University of Illinois in France. We will discuss about mixed focused ultrasound microelectrode array platform for spatial-temporal description of first elicited local field potential in an ex vivo marine hippocampal structures.
Thank you, Aputu, for the introduction. And my name is Ivan Castellanos, and I am very happy to be here attending FUN22 conference in Germany with you all. And before I get into the experimental details of this work, I'm going to briefly go over the motivation for this study, which is kind of the reason why we're all here in the first place. It is the burden of neurological disorders. And in a recent Lancet study, it was estimated that between 1990 and 2016, neurological disorders represented the leading cause of disability-adjusted life years, or DALYs. There's a public health metric to measure the number of years that have been lost to a given disease. And over here we have a map of the distribution of these DALYs. The distribution of these DALYs across the world. It is also the second leading cause of deaths worldwide. And unfortunately, this burden is expected to continue increasing over the next years because of an ever-increasing and aging population, which has also increased the demand for treatment, rehabilitation, support services to address these disorders. In the case of neurodegenerative disorders, the first line of treatment is pharmaceutical strategies, which may come with many undesirable effects and can lose their effectiveness over time, which led to the development of neurostimulation technology. And neurostimulation technology, as you may all know, comes in many forms. The most widely used is deep brain stimulation, which consists in the surgical implantation of an electrode at a given location of the brain, and the sending of electrical pulses to stimulate it. There are other transcranial techniques, non-invasive transcranial techniques that use magnetic fields, such as transcranial magnetic stimulation, and direct and analog current stimulation that utilizes electric fields to modulate the neural activity of the patient. However, these technologies are often confronted to a trade-off between the high spatial selectivity that they can offer and the invasiveness of the treatment. This is why I believe we're all fans of focused ultrasound. It is because we can, in theory, we can apply it non-invasively, and we can focus and we can have high spatial selectivity since we can focus the ultrasound energy into specific regions of the brain in the cubic millimeter range. On top of that, we could implement beam steering to treat different regions of the brain in one treatment. So this is the reason why the neurostimulation project at the Labto was created, and the main goal is to study the biophysical and electrophysiological mechanisms involved in neuromodulation or neurostimulation by ultrasound. And to do so, we developed several platforms that use different neural models, from in vitro models to ex vivo models to in vivo models. In vitro models, we have results and experiments performed on neural cell cultures, and I would like to invite you to stop by the poster of Tomo Vie, who's a PhD student in our lab, and he can tell you all of the great work he's done in his first year of a PhD in this model. We have also an ex vivo model that uses microelectroarrays to measure the electrical activity produced by ultrasound stimulation on hippocampal brain slices from mice. And we also have several in vivo models that range from the simple common earthworm all the way to more complex murine models and porcine models. However, today I will present the results that we've obtained using the microelectroarray. The purpose of the study was to demonstrate the feasibility of using both microelectroarrays and focused ultrasound systems to investigate the neural signals generated by single pulse focused ultrasound stimulation, and therefore evaluate the causal effect of the stimulation. We also wanted to describe the spatial temporal characteristics of these responses. So for those of you who may not be familiar with microelectroarray systems, these are systems that allow for special temporal recording of electrical signals in brain slices with very high temporal resolution of approximately 50 kilohertz. On the top right corner, we see a microelectroarray chip with a hippocampal brain slice from mice. These chips contain, or the chips that we use, contain a grid of 60 electrodes spreading every 200 micrometers, as you can see in the bottom right corner. And the first thing that we do is to place the hippocampal structure on top of the electrode grid. Once we do that, we identify the major neural pathways of the hippocampus, mainly the bossy fibers and the Schaefer collaterals. And it is the Schaefer collaterals, the main neural structure that we'll be studying. So you'll be hearing that word a lot. So these systems allow for recording of action potentials from multiple neurons, also known as multi-unit activity, as well as synchronized synaptic potentials, also known as local field potentials, composed of fiber volleys and field excitatory postsynaptic potentials, and I will go over what those mean a bit later in the presentation. But this is the type of data that we can obtain using the systems. As you can see, it is a matrix of neural electrophysiological signals recorded at different sites within the hippocampal structure. So now I'm going to briefly go over the experimental setup that was used for these studies. We have a schematic on the right and a picture of the setup on the left. As you can see, the focused ultrasound transducer is coupled with the artificial cerebrospinal fluid that is constantly perfusing the hippocampal brain slice via a coupling cone that was filled with agarose gel, and that will transmit the ultrasound beam to the location of the hippocampal brain slice. Here is a close-up of that integration between the transducer and the MEA chip. And to position the exit of the cone, and therefore the arrival of the ultrasound beam, we used an integrated camera that comes with a microelectroarray, and we positioned the edge of the cone, or the rim of the cone, to be directly over the MEA grid, and therefore we know that the ultrasound beam will be directed to the hippocampal structure. So the first set of results that we wanted to obtain was a direct comparison between the responses generated by electrical stimulation and those generated by focused ultrasound stimulation. For electrical stimulation, we selected an electrode located in the vicinity of the Schaefer collaterals, more towards the CA2 region of the hippocampus, and we applied a biphasic pulse, much like this one. For ultrasound stimulation, we tried to aim the ultrasound stimulation and target to the CA3, CA2 region of the Schaefer collaterals. And we applied single focused ultrasound pulses of 160 microseconds, pressures up to 6.3 megapascals, and a frequency of 1.78 megahertz. And for the recording electrode was located downstream the Schaefer collaterals. Downstream the Schaefer collaterals. and we recorded the activity produced by both ultrasound stimulation and electrical stimulation. And when we get the results of these responses, we have the ultrasound stimulation responses in red and the electrical stimulation responses in blue. We can see that they follow similar patterns in that they both start actually with a stimulation artifact followed by fiber volleys. Fiber volleys are indications that the electrophysiological signal or the action potentials have reached the synapse of the axon. And those negative deflection fiber volleys are then followed by a positive deflection known as the field excitatory postsynaptic potentials, which are indicative of the transmission of the electrophysiological signal from the synapse to the postsynaptic neuron. So now that we identified the responses generated by focused ultrasound in time, we were interested in seeing how it was distributed in space. And therefore, in this diagram, we see a reconstruction of the voltage recorded by all 60 electrodes in response to a single pulse focused ultrasound stimulation using these characteristics. Over here, we have a temporal signal recorded in electrode 23. The electrode was selected to be located, again, near the Schaefer collaterals, downstream in the CA1 region. We aimed the ultrasound stimulation in the CA3 region. And when we run the movie, we see that we first noticed that there's a very strong negative voltage recorded by all electrodes in the MEA, and that corresponds to the focused ultrasound artifact. If we continue running the movie, we're gonna find that at least in electrode 23, that is when we're gonna detect the fiber volley. Again, the arrival of the action potential to the synapse. And finally, when we take a look at the distribution of the field excitatory postsynaptic potential, when the signal is transferred to the postsynaptic neuron, we see that there is an accumulation of strong activity in the CA1 region after stimulating the CA3 region, meaning that the signal was not only generated, but also transmitted along the Schaefer collaterals and measured at distance in the CA1 region. And after we continue running, we see that we have a slow and progressive return to pre-stimulation baseline. So again, once we defined the spatial temporal characteristics of these responses, we wanted to study the effects of varying some focused ultrasound parameters and to see how if they have any effect on the responses that were measured. We have our matrix of signals over here, and again, the identification of the major neural pathways, the massive fibers, and the famous Schaefer collaterals. We selected an electrode in the vicinity of the Schaefer collaterals to study the responses generated by ultrasound at different pressures, actually. And when we apply pressures of 3 megapascals, we see that the only thing that we can measure or observe is the stimulation artifact. If we increase to 4.1 megapascals, we start observing the emergence of a postsynaptic potential. We further increase the pressure to 5.3 megapascals. We see that the amplitude of the postsynaptic potential has increased. And finally, when we increase the pressure to 6.3 megapascals, not only do we further increase the amplitude of the postsynaptic potential, but if we take a closer look at the beginning of the response, we see that there is also an emergence of the mossy fiber, which indicates that we weren't only capable of generating neural responses in the Schaefer collaterals, but that those neural responses again were transmitted along downstream this neural pathway. So in conclusion, we introduced a mixed focused ultrasound microelectrode platform as a viable tool for studying the spatial temporal characteristics of focused ultrasound neurostimulation. We showed that single focused ultrasound pulses are capable of stimulating and transmitting responses down major neural pathways, at least in the hippocampal structures. And in future studies, we will continue our parametric studies to see how varying different parameters can have an effect on the measured responses. And we will continue doing studies to identify different biological and cellular actors in neurostimulation and neurotransmission by focused ultrasound. With that, I'd like to thank everybody involved in this project at the Lab Dome in Lyon, France. as well as our collaborators in Paris and our funding agencies, the Focus Option Foundation, the French ANR, and the LabX that we can. With that, thank you for your attention, and I will now be happy to take your questions.
Thank you very much. Any questions? Yes.
Did I see correctly that not all of the NDA collaborators responded? And if so, have you quantified between how many slices, how interaction is responsive to government?
Yeah, so it is true that when we send the consecutive pulses, it appears that only like a certain region is activated, and I think we believe that normally it's supposed to be the region that is innervated by the, or that includes parts of the Schaefer collaterals. We haven't actually measured actual areas to and see how much those the area of activation varies between pulses, but that is a great suggestion. Between slices in our study that we published in the Journal of Neural Engineering, we used six slices. It was pretty reproducible, but we did see some differences in the amplitudes of the postsynaptic potentials, and also the duration of the after we reach the peak of the postsynaptic potential, the duration by which it descends to pre-stimulation baseline. So just different characteristics, but in general pretty reproducible.
I'll speak up.
Yeah, I hear you. Yeah.
I was wondering if you could comment on the dimensions of the sonication field.
Yeah. Yeah, of course. And it's it is true that with respect to the hippocampal structure, it is quite a big focal spot that we're using, and I guess FUN22 likes to use the metaphor that we're like basically using a hammer to go and hit those structures. So we're like in a way we're not as precise as we would like to be, but the good thing is that with such a high such a big focal spot, we are pretty certain to be hitting the regions that we want to aim, which is the CA3 region of the hippocampus. But now, like I guess this is part of future work, and actually work that is under development, and that is like the first steps of that development are actually included in Tomo V's poster, is that we're trying to like use higher frequencies to minimize the focal spot and try to have a better accuracy, better targeting, and study those effects as well.
Thank you very much again. So I forgot to mention that it's a technical problem we have with the previous presenter. We need to replacement of this presentation by Julie Cannon from the neuroscience session, so we will switch to presentation. So this is not the last presenter now. Okay. But no, you will present now. But, so now I'm happy to introduce Dr. Theo Lemaire from New York University School of Medicine, and we'll be talking about direct two-photon imaging of focal and cell type selective ultrasound neuromodulatory effects in the mouse cortex. Thank you.
So hello, everyone. It's a pleasure to be here and to reunite with the ultrasound community in person. I am Theo Lemaire, a postdoc in the Neural Interface Engineering Lab of Professor Shai Shoham at New York University. And today I'll be discussing with you our efforts to decipher ultrasound effects in vivo at the cellular level using the mouse cortex as a model. So let's dive in. why do we need to understand ultrasound effects directly at the cellular scale? And the reason is twofold. On the one hand, ultrasound neuromodulation is associated with a multidimensional parameter space, where any given stimulus is characterized by its carrier frequency and pressure amplitude, or alternatively the pulse intensity, and also its pulsing parameters, so burst duration, PRF, pulse duration, or alternatively stimulus duty cycle, its spatial focus, and also more recently, the nature of the waveform itself. And this virtually infinite space allows for great flexibility, but it also leaves us with a complex optimization problem. And on the other hand, I don't need to stress to this audience that ultrasound neuromodulation can elicit a large variety of brain-wide effects, including both activating and suppressing effects, sometimes reported within a single study. And the parameter dependence of these effects is not yet fully understood. Part of that knowledge gap stems from the fact that almost all of the studies in our field have looked at indirect effects of ultrasound neuromodulation, such as elicited motor responses, EEG and fMRI functional signatures, and behavioral effects. And the indirect natures of those readouts limits our ability to understand direct ultrasound neuron interactions and their parameter dependence. In contrast, a lot of recent work in systems neuroscience has focused on trying to understand the effects of neuromodulation at the level of cell types, for instance, with optogenetics. And this work has led to a greater understanding of the macroscale effects of neurostimulation. So inspired by this research, we asked, what is the direct impact of ultrasound stimulation on the local neural circuitry? And to answer this question, we defined three main objectives. First, to develop a 2-photon microscopy platform combined with tightly focused ultrasound neuromodulation in awake mice. Second, to determine the effect of stimulation parameters on neural activity in the cortex. And finally, to investigate the cell type specificity of these effects. And I'm going to start by discussing our experimental platform. And at its core, we have a conventional 2-photon microscopy setup comprised of a femtosecond laser emitting excitation red light, a high numerical aperture objective focusing the light on a small target, and a photomultiplier tube that picks up the emitted fluorescence at a specific wavelength of interest connected to our acquisition setup. And beyond the objective, we placed a transgenic mouse expressing a fluorescent reporter in cortical neurons and surgically implanted with a cranial window, allowing us to image its cortex. And I want to point out that this setup allows us to image from different layers of the cortex by simply moving the objective up and down. And finally, we coupled our ultrasound drive chain to this setup that connects to a ring transducer that you can see here that allows us to image. It is affixed to the objective, but it allows us to image through its central aperture. And this transducer has a lateral footprint of about 1 centimeter and the central frequency of 2.1 megahertz. And as I'll show you later, it produces a tightly focused beam while still stimulating the entire field of view. And with this platform, we could acquire fluorescence videos showing the functional response of cortical neurons to an applied ultrasound stimulus, such as this one, where you can see a widespread activation of cortical neurons upon stimulation. Speaking of stimulation, we vary 2 critical stimulation parameters, so pressure amplitude from 0 to 0.4 megapascal peak amplitude, and the stimulus duty cycle from 5 to 80%, while keeping other parameters fixed. And in order to analyze this data, we started by denoising the fluorescence movies, and we then fed them into this Sweet2P analysis pipeline, which is a standard in the world of 2-photon microscopy. And what this pipeline does is it first aligns the movie frames to compensate for motion correction. It then identifies and segments neurons within the field of view and extracts fluorescence traces for these neurons. We did then was to normalize the fluorescence traces according to their baseline in order to bring them to a scale that is comparable across neurons, animals, and sessions. We then rejected outliers that showed either significant plane motion or abnormal pre-stimulus activity levels. And we then characterized the response of each cell to each ultrasound condition by averaging across repetition and just performing a statistical test to assess whether there was a response. Finally, we characterized the responsiveness of each cell by just counting the number of conditions it responded to, and we derived summary statistics for each cell category. So this process data allows us to better visualize the impact of an ultrasound stimulus on the activity of each individual neuron identified in the field of view. For instance, for this data set of about 400 neurons, we can see a very robust activation of pyramidal neurons due to the ultrasound stimulus. And we can go a step further and look at the pressure dependency of these activations. And what we see here is that there is a very clear threshold and the cells start getting activated above some specific pressure level. And we can also look at the duty cycle dependency of these responses. And what this shows is a clear increase in response magnitude with increasing duty cycles. So one obvious concern here is the question of the focality of the pressure field and the evoked effects. So to quantify this focality, the first thing we did is to look at the pressure field generated by a ring transducer. And our mapping reveals that we have an actual focus of about 1.5 millimeter that allows to selectively target the mouse cortical layers without impacting deeper regions. And we have a lateral focus of about 0.5 millimeter that corresponds to the size of our field of view. To verify this focality functionally, we characterized ultrasound evoked responses while introducing a progressive lateral offset between the transducer and the objective. And indeed, responses are highly focal and they decrease dramatically even when we move the transducer by only 0.5 millimeter. So having verified that we could elicit robust and local effects, we set out to analyze the responses evoked by ultrasound in different neuron types. And specifically, we looked at 3 classes of cortical neurons, each identified by specific neurochemical markers. We looked at Taiwan positive pyramidal neurons that constitute the primary excitatory unit of the cortex. We looked at somatostatin positive inhibitory interneurons that contain the low threshold spiking cells and that target excitatory dendrites. And finally, we looked at parvalbumin positive fast spiking inhibitory interneurons that target excitatory neuron cell bodies. And to image from each cell type separately, we use separate transgenic mouse lines, each expressing the calcium indicator GCAM6S in using a cell type specific promoter. And I want to stress on here that these 3 cell types together, they constitute a significant proportion, over 90% of the total cortical neuron population. Right, so what did we find? With excitatory neurons, we found that about half of the population shows robust ultrasound responsiveness. And in terms of pressure dependency, these neurons start responding around a threshold of 0.15 megapascal, and then show a monotonic increase in response magnitude with increasing pressure levels. Complementarily, they show a more complex dependency on duty cycle with a peak in responsiveness around 50%. The SST interneurons, we found that almost the entire population showed robust ultrasound responsiveness, and these neurons display a very similar pressure dependency as the excitatory neurons. And they also show a peak of responsiveness at 50% duty cycle, as you can see here. But interestingly, there's also a sharp increase in responsiveness between 5 and 10% duty cycle, which I'll come back to later. Finally, for the parvalbumin positive inhibitory interneurons, we found that only a very small portion of these neurons responded to the ultrasound stimulus. And these neurons were only excited at the highest pressure level, as you can see here. So these results provide strong evidence for a pronounced cell type specificity of ultrasound evoked effects. So I'd like to zoom in now on one of the most interesting features of these results, namely the differential excitability of pyramidal neurons and SST interneurons across duty cycles. So comparing the evoked activity of these 2 populations, what we notice is that at 5% duty cycle, neither population shows strong activity. But then at 10% duty cycle, the SST interneurons start responding robustly to the ultrasounds, while pyramidal neurons stay mostly silent. And then at 30% duty cycle, both populations show pronounced activity. So if we recall the physiological function of these 2 populations, these differential activations across duty cycles could be a driving factor to either suppress or enhance cortical activity across these duty cycles. And this feature is consistent with theoretical predictions from our group, but it's also consistent with other theoretical models out there. So before concluding, I'd like to put these results in perspective in light of other recent studies that also looked at ultrasound effects at the single neuron level. On the one hand, I selected 2 studies that are also looking at ultrasound effects in the mouse cortex with 2-photon imaging. A study from the Hong Chen group that used a similar frequency and higher pressure levels, and that found that ultrasound did not elicit any response in pyramidal neurons. And a study for the Meng Kui group that used a much higher frequency and found that ultrasound suppressed neural activity. So our results clearly depart from these observations that we show ultrasound-evoked neural activation very robustly. And on the other hand, we have another 2 studies that, like us, reported excitation. One from the Xue group using 1-photon imaging and showing that ultrasound induces strong activation in both hippocampus and motor cortex neurons, but lacking cell type specificity. And another from the Binhe group showing using electrophysiology to classify spiking units, cortical spiking units, and showing that ultrasound evokes distinct effects in different cortical spiking units. But this technique also does not allow to differentiate between pyramidal neurons and SST2 neurons because they are both regular spiking units. So our results complement these studies by looking more robustly at the cell type specificity of effects. So in summary, we developed a platform allowing single neuron functional imaging in a scaled model of mammalian functional focused ultrasound neuromodulation. We showed that ultrasound elicits robust and local responses in cortical neurons. And we showed that ultrasound evoked responses are dose-dependent and cell type specific. So to wrap up, I'd like to thank contributors to this project. Of course, Professor Shai Shoham for his mentoring and guidance throughout the project. Postdocs Yuan and Justin Little, who spearheaded the development of our experimental platform, and our collaborators Emile Massurier and Professor Rob Fromke from NYU for their helpful collaboration. All the Shoham lab, and in particular the trainees who took part in this project, Juni Hook Lee, Ben Stetler, and Sarah Heiken. And finally, I'd like to acknowledge the NIH and the Swiss National Science Foundations for sponsoring this work. And with that, I thank you for your attention, and I'm happy to take questions.
Any question? Yep.
When you change duty cycle, do you change the amplitude, the pressure
No.
as well? Okay, so when you change duty cycle, you're delivering more ultrasound. Okay, thank you.
Yes.
I did try 100% duty cycle.
No, we haven't. The maximal value we tried was 80%. And we explored duty cycle with a fixed pressure amplitude value.
Yes.
Very one. You mentioned that you bring the to show how focal the is. If you observe while in that, like the delay
So I can try to go back to the slide, but I don't think we observe any strong delayed responses. And also, I think part of the reason for that would be that if there were, they might not be that synchronized as the direct ultrasound elicited response.
Okay.
Can you speculate why what underlies the cell type specificity?
Sure. There's several theories out there. One of intramembrane cavitation that our group is, I guess, famous for, that states that indeed these low threshold spiking cells have like specific channels that allow them to sort of like accumulate charge also during off periods, and this might, sort of provide them with the ability to respond also at lower duty cycles. But this is not entirely specific to the intramembrane cavitation model, because these neurons actually they do have these channels, and these channels could be, this is what differentiates them from also other neuron types in terms of membrane composition. So probably this T-type calcium channels would probably play a role in this.
Yes.
I'm mindful that it might be a very unfair question, but you're very compelling results of local activity. But then when we apply stimulation at a system level, we don't see an evolved response. Can you speculate on why that might be the case?
You mean, can you,
Like a motor evoked potential or very, low speed, that
I mean, I don't see how these results would be incompatible. If your focal size is like large enough, then you can clearly induce a motor response if you're sonicating over the motor cortex.
Maybe I think of it as conversation. So, we showed that there was a, 20% of these levels, how to reflect, 30% of the strategy. Well, when there's 10% of the reflect, the need to go on 30% of the strategy.
So this one,
I think so,
yeah. So 10% we see mostly activation of these inhibitory interneurons, the specific population of interneurons, and then 30% we see both being activated.
Okay, thank you very much again.
Yeah, did you look at like,
Oh, sorry, but we're running out of time, so we can continue. Thank you. Thank you very much. Okay, so the last presenter is coming from the next session, actually. Session. So Tuli Kanandi is working at the Neuroimaging Center of UNS Gutenberg University. And she's going to be talking about disentangling transcranial and auditory sources of transcranial ultrasonic stimulation induced suppression of corticospinal excitability.
Thank you. So I'm going to talk about how we could try to look at what effects we get from ultrasound itself, and potentially some things that might be caused by the auditory artifact that accompanies the pulsed ultrasound that we apply. This work was done here in Mainz in Till's lab with support from a number of other people that you will see here today. So this is just to show that there's at least a couple studies and more now, which have shown that when you apply online ultrasound for say 400 to 500 milliseconds, and then measure the motor evoked potential using TMS right at the end of that, you see a suppression of the MEP. So in this graph, you will see that, oh, you don't see my pointer, never mind. The darker gray, which represents the ultrasound condition, the MEPs are smaller. And then in this other study, the same thing was examined, but now across many different TMS intensities. And again, you see now in lighter gray, the ultrasound has smaller MEPs. But we know that there is an auditory confound that accompanies the ultrasound that we apply. If you have placed your transducer in a water tank and stood next to it, depending on the transducer that you have, you might be able to hear the tone when the ultrasound is on. The question is, why is ultrasound audible? And what we think is that because for most of these human studies, we've applied ultrasound in a pulsed manner. So we turn it on and off. And for these pulses are repeated at a pulse repetition frequency, which is often in the audible range, which is why we are able to hear it. And this is a study which shows, so without going into the details, on the y-axis of all of these graphs, you have the percentage of responses that were audible. So when it is very high, people were able to hear all the ultrasound trials. When it is low, they were not able to hear as many. And what we've done in this study is we've either applied a mask on top or tried ramping the ultrasound. So when we pulse it, instead of turning it off and on in a rectangular fashion, if we ramp it up and down, then it might be less audible. But the question is, do we really care if it is audible? Is that a problem for any of our studies? And this is a study in rodents where I will focus on these two. What you're seeing is EMG recordings. And you can see that both ultrasound and audible sound are having a very similar response. And in fact, the air puff right on top is also having a similar response. So what these people did was they used a model where, in this case, in orange, you have deaf mice. The blue is sham, meaning these mice can still hear. So the deafening procedure was sham, not sham ultrasound. And when you look at that again, you will see that the sham mice in blue have an EMG success rate that is pretty similar for sound and ultrasound. But when they are deaf, both of them respond less. And interestingly, the coupling, which is whether you have a good gel medium to between the head and the transducer, that seems to affect the ultrasound response, which becomes much smaller in the absence of coupling. One possible solution that I already mentioned is to apply an auditory mask. And here's one study which did that. In maroon, you see the ultrasound burst, and then time locked to the ultrasound, they apply an auditory mask, which is at 1 kilohertz, which is the pulse repetition frequency. So they use the square wave. This is the same wave or same pattern as the envelope that we apply to get pulsed ultrasound. And in this graph on the y-axis is the probability that someone was able to detect that a trial was ultrasound versus sham. And what you see in the mask condition is that the probability is much lower. So there's a few participants who can still tell the difference between sham and ultrasound, but most of them can't tell the difference anymore. The other solution that is possible is the ramping that I mentioned. So in this, the black traces are the auditory brainstem response. And you see that occurs when there is a rectangular pulse at the beginning and end of this sharp change in intensity. But in the lower right figure, where it is ramped up slowly, then you don't see the brainstem response anymore. The sound, we think, is partly conducted by bone conduction. And that's what is shown in this modeling study, where they show that there was a lot of displacement in the inner ear in the red line, which is the sharp rectangular envelopes, but not so much in the ramped condition. But there's also likely some bone conduction occurring. And it is possible that because some of this is bone conducted, applying the masking via bone conduction earphones might be helpful. So that's what we did in this study, where we basically tried to develop an auditory mask so that participants would not be able to hear the ultrasound well. What we have is we had two targets, one in the primary motor cortex and one in the deep white matter, which was our control condition. And we had that either with the sound on or with the sound off in both of those targets. And we then looked at this at 3 different intensities of ultrasound. What we hypothesized first was that MEPs would be suppressed by the ultrasound only when the target is M1. So if the auditory confound doesn't have any effect, then we should see suppression only when the anatomical target is hit by the ultrasound. We also hypothesized that there might be lower suppression in the presence of the auditory mask, suggesting that some of the suppression may be due to the auditory mask. And finally, we expected higher suppression at higher intensities. For this study, we used the NeuroFuzz 250 kilohertz transducer, and we paired it with TMS. So the transducer is under the TMS coil that you see in that picture, because the transducer has to be directly coupled with the head. And of course, we had EMG, so we could measure motor evoked potentials. And the whole thing was neuronavigated using that tracker that you can see on the TMS coil. The protocol that we used, 250 kilohertz, 1 hertz pulse repetition frequency, and a 30% duty cycle, which is what has been shown to cause suppression of MEP in previous studies. We had 400 millisecond trials, and at 390 milliseconds, we applied the TMS pulse. So we had some trials where we only had TMS, and some trials where we had ultrasound that preceded the TMS. We coupled the transducer to the head using a combination of gel and a gel pad on top of that. The masking sound that we applied was a square wave at 1000 hertz with some white noise added to it. Now, If you've not heard this sound before, the 1 kilohertz is quite an annoying sound. It's very high pitched and it can be annoying for participants. And adding the noise seemed to make it a little bit more comfortable for them. We applied this via bone conduction earphones, and the sound was played continuously. So unlike the study that I showed where the mask was time locked to the ultrasound stimulation, here this just played throughout, and then sometimes you would have an ultrasound with a TMS pulse or a TMS pulse by itself. And for each participant, we increase the signal to noise ratio and the volume until they could either no longer hear the ultrasound or it was very uncomfortable for them. So we had a total of 12 participants after excluding a few, because when we have the transducer below the TMS coils, sometimes it's too far from the cortex to get an MEP consistently. And what we find here, I'll walk through this graph one part at a time. The first two columns that you see are when we apply the ultrasound to the M1 target. The first one being without any mask, and then the second column with the mask. And then the next two columns are the same thing, without and with a mask, but now targeting an area deeper in the bone. Each of the three rows are different intensities. And what you see on the y-axis is the size of the motor evoked potential. So this is motor evoked potential in the presence of ultrasound as a percentage of MEP in the absence of ultrasound. So the black dotted lines reflect 100%. Any data points below that show suppression of MEPs. Anything above that shows excitation of MEPs. And first we ran a one sample t-test to check whether there was any suppression in any of the conditions. And we found that all the conditions, except the two that I have highlighted here, which are the ones, which happen to be ones that had a mask applied, we do see some suppression of MEPs. This is preliminary, so we also wanted to run a Bayesian ANOVA to look for whether there is some sort of trend for the effect of the mask, and we use the target mask and intensity as factors in the ANOVA. And we find a base factor of approximately 16 for the mask, suggesting that there is lower suppression in the presence of the auditory mask. And if you look at the top left there, you might see this a little bit where you see greater suppression in that first graph there, but lower suppression in the next one right next to it. We think that it is possible that the MEP suppression is in part due to the auditory co-stimulation, but we had some major limitations here. And one of them is that our mask was not effective for all participants. Sometimes when we tested at the beginning, participants said they couldn't hear it, but then later on during the testing, they said, now we can feel it. And if any of you have had it on yourself, I've tested this a lot on myself with different masks. The ultrasound sounds distinctly different, possibly because of the way it's conducted through the bone. For me in particular, it sounds like the ultrasound is moving from one side to the other. So if I have a static mask in the background, I can still tell that the ultrasound is different. But this is different for different participants. Sometimes you put the transducer on one side of the head and the participant says, I feel like the sound is coming from the other side. So it's fairly complex is what I'm trying to say. And it's not very easy to mask by just applying a sound in the background. One of our other major limitations is that we use the same focal depth for all participants. And here you will see a little red line coming up. And on the left, you will see that ends up around the hand knob. But on the right side, it ends up a little bit deeper. And based on the talks this morning, you might already probably know that there's also some defocusing, which we haven't taken into account here at all. In our next phase, we want to improve the auditory masking, individualize the targeting, and potentially also go back and do some simulations on the data that we already have to see whether we were actually hitting the target. And to also add a positive control where we want to see what happens if participants just hear a sound. Does that by itself cause a suppression of the MEP? There's also other possible solutions, which I alluded to very briefly. And if you would like to hear a bit more about ramping, please come to my poster. Thank you.
We take time for a few questions. Yes.
So, let's look at one comment. So it looks like, and correct me if I'm wrong, but it's the first application of descending to results, and all and I think, and the comment is, I think it would go a long way to block the community to see that application. All the time. I think this will go a long way towards addressing some of the skepticism out there about the biological effects. So
just a comment on that. What I should have said is that I'm not implying that the ultrasound doesn't have an effect. You will see a lot of evidence of that over the next couple of days. But what I'm saying is that in some experimental setups, you need to be aware that some of the effect might not be because of the ultrasound directly.
Yes.
Congratulations on the nice work. It seems like there are a lot of bias for the field in terms of some technology.
So that was not intentional. This is a little bit of a convenience sample here, but I wouldn't know what to expect if we looked at differences.
Other question from the assembly? No?
No.
Yeah, maybe it's lunchtime now, so we can continue the discussion with you at lunch. Thank you very much. We have a discussion, which was a mixed session. So let's go to lunch, and the third session will start in about 1 hour. I think 1:15. So let's